In this lesson, we shall talk about DNA fingerprinting. Now, what is use of DNA fingerprinting? How it was discovered? What principle it utilizes? Okay. So, as the name suggests, you have to find out the fingerprint of DNA content of an individual. Now, you might have heard about this term fingerprints in some murder mystery, or if you're watching or reading some sort of murder scenario, fingerprints of the criminal are to be traced out. In the same way, the fingerprints of DNA, that means DNA composition of an individual, when it is charted out, that is known as DNA fingerprinting. As the fingerprints of your hand, they have the usage in tracing out the crime scene's criminal. In the same way, DNA fingerprinting is also used in those criminal scenes and as well as disputed parentage cases where the parallel has to be drawn between the sample's DNA and the convict's DNA, and then that has to be matched. Now, one thing you know by now, that this being the last topic of genes, you know by now that every individual has a genome which is totally different from the other individual. One thing that is clear. Secondly, you know that human genome or the human base pairs, the DNA, which is having as many as 6 billion base pairs inside it. To cut it short, even if we consider the gene, there are 3.3 billion base pairs, which is a huge number. And one more thing came to our knowledge from the Human Genome Project was that 99.9% of the genome is similar in all individuals. Now, the difference lies in 0.1%. Now, it is not segregated in such a way that 99.9% of DNA is similar, so that would be present similarly and the remaining 0.1 would be present at some end, and you will get to know that what that 0.1 difference is. That 0.1% difference is present within that 99.9%. Now, imagine if you have to draw parallel between DNAs of two individuals and you have to find that 0.1% difference out of that 100%, which is 3.3 billion base pairs. How daunting task that would be. What a big task it would be. So, for that purpose, it is better we seek that region which is easy for us. I am going to repeat all what I have said. You get an idea why we are looking for DNA fingerprint of an individual. See, supposedly this is 1.1 meter of DNA of an individual. It is having this composition, okay? And the other individual is having this DNA. Now, there is one statement that 99.9% of the DNA is similar, as I have tried to present over here. Now, that 0.1 difference lies either over here, here. Rest all is similar. This is the point of difference, 0.1 difference. And in the other individual, it would lie over here. Like this, right? Now, for finding out where the difference lies, you have to encode this entire one and this entire as well. And this in entirety is 3.3 billion base pairs. A huge thing. It took 10 years to encode this completely. How difficult it would be to encode for every individual. So, it is better to find the answers not in that 0.1%, but in that 99.9%, okay. So, we see the answer, that is, if we have to trace some similarity or difference, we will find that similarity or difference in the repeated sequences. Most of the part of DNA has repeated sequences, and those repeated sequences will be the place where we are going to find the information and use it for DNA fingerprinting, for matching, okay. We are going to see the similarities in these repeated sequences and whatever differences, if there are any. Now, apart from seeing this entire genome, we are going to concentrate on a smaller part, and in that smaller part we have a principle known as DNA polymorphism. Now, to understand DNA polymorphism, I would take you back to the fundamentals of genetics. Few things would come and crop up from there. So, you can watch that video all over again, and if you have that thing fresh in your mind, that's quite good. What DNA polymorphism means? We had talked about SNPs. We said that there is something known as single nucleotide polymorphism. We had talked about repeated sequences. In the DNA, there are a lot of repeated sequences, as many as 10,000 times repetitions are taking place. Now, within those repetitions, there could be a difference or there could be a similarity. Now, what do we see in DNA polymorphism? So, basically, this DNA fingerprinting lesson that you are having over here can otherwise be considered that we deal with this term properly. We are going to see what DNA fingerprinting is all about, how it takes place, what all is carried out in the lab, but this lesson is about DNA polymorphism. So, brace yourself for understanding what it is. First thing, that repeated sequences are there in the DNA. 99.9% is similar in all individuals. Out of that, we are taking up the repeated sequences where the repetition is of great number. In that, we have single base pair polymorphism. Single nucleotide would be different in all those sequences, and this difference we are going to target, okay, when we have to do fingerprinting. Now, how this DNA polymorphism comes into scenario? First of all, you have to keep in mind that when the DNA is separated, it is being cut from the chromosome or the entire genome. There are two types of DNA. One is bulk DNA and other one is satellite DNA. When this DNA is separated, there are two peaks which are made. The one which is satellite, that makes a minor peak, while the one which is the bulk, that makes the major peak. So, you have a minor peak being formed and a major peak in the centrifugation. Okay. Now, depending on the types of repetitions and how they arrange themselves, these satellite minor peaks, satellite DNA could be mini satellite, microsatellite, the satellite DNA varies. So, we have a satellite DNA that is forming the minor peak. Now, this satellite DNA has repeated sequences. It is of no use, it just codes for repeated sequences. In these repeated sequences, we find polymorphism. Now, this polymorphism is just like this. Supposedly, you have a sequence A, T, T, G, G, C, A, T, T. This is one sequence. A similar sequence is being repeated. Only point of difference is that at place of G, you are having C. Then you have another repetition of the same thing. At place of C, you are having G. And this is repeated many times. This would form the satellite DNA. Now, this polymorphism has come as a result of mutations. See, when we studied mutations, we came to know that the mutation, if it is inherited, if it is present inside the germ cells, an individual undergoes many mutations during his or her lifetime, and if they are somatic, they are not inherited, but in case the mutation comes in the germ cells, that would be inherited. Now, in case that mutation comes in these non-expressive sequences of the germ cell, it would be inherited and it will not have any effect on the phenotypes, okay. Then moving further, the offsprings would inherit it, then it would go further and further, and from the idea of allelic frequencies that we talked about in Hardy-Weinberg equilibrium, if the frequency of a mutation is 0.01 in the gene pool of a given population, it is nothing but it is polymorphism, okay. If the mutation is being seen up to this much frequency in the gene pool of a particular population, that means allelic composition is showing this much variation, then it forms the polymorphism. Then we say it that it is DNA polymorphism. I hope you get an idea that in the germ cells, if some mutation takes place in these sequences which are not expressing themselves in the form of genes, those mutations will not be expressed, they would be inherited as such. Their inheritance goes from one generation to another because the offspring takes up those sequences. Now every individual is going to inherit some or other sort of mutation that their parents have inherited in their lifetime. So these repeated sequences and the polymorphism that occurs in these repeated sequences would vary from individual to individual, and we are going to seek the polymorphism with such an allelic frequency in the population that we discussed. We are going to seek this polymorphism as the main point where we are going to find the similarity and differences and use it for DNA fingerprinting. Alright, so this is what DNA polymorphism is all about. I am going to repeat what all I have said so that you get an idea. If I could present it in other words, just to make you understand. It is little bit complicated because we bring back those fundamentals of genetics as well over here. See, whenever the DNA is separated, it forms two DNAs. One is bulk and other is satellite. Bulk forms the major peak in the centrifugation while the satellite forms the minor peak. Depending upon how they are further classified, we have mini and micro satellites. That depends on another methodology. We don't get into depth of that. The minor peak DNA we are going to take up. Now, in that minor peak DNA, we find DNA polymorphism. This minor peak is formed by that DNA which does not express itself and it has repeated sequences. These repeated sequences, they have polymorphism. Now, what is polymorphism? Because of mutation in the germ cells which do not express themselves, they are inherited one by one after generation and generation, and they form that allele's frequency this much. If it is this much present, then in the gene pool it is known as polymorphism. Basically, it is a mutation, it is a polymorphism that if inherited from one generation to another and contributes as much as this much value in the gene pool for an allele, for a given allele which does not express itself, then that is known as polymorphism. Don't get confused that we are using the term allele, so alleles are for genes. Over here, allele is simply a sequence, okay, that would be coding for something, but it does not have an expression. So if the allele is having this much frequency in the gene pool, that is known as polymorphism. That means it has shown its effect in the population. When it has shown its effect in the population, that means the genetic content of that individual is different from the other individual. So, like your fingerprints are different from the other person, in the same way some part of your DNA is different from the other person. He or she would have inherited it from his or her parents, and that would be totally different from what your parents gave you. So in a way, you get to think that fingerprinting of DNA content of repeated sequences will make sure that whether there is some similarity at the genetic level or not. And we are going to talk about the applications, but for now I want you to understand what this DNA polymorphism is all about. We are going to target this DNA polymorphism inside the lab. We are going to trace the similarities in that polymorphism and figure out who was the criminal. So this is what DNA fingerprinting is all about in terms of DNA polymorphism. How it is done, that is part of next lesson.